pick up where I left off last time, of course, and we're going to start talking about myocytes. We're going to talk about these cells that make up the heart. And we're going to see that myocytes come in these two great categories. The cardiomyocytes, the muscle cells of the heart, the cells that make up the myocardium, come in two varieties. One of which is said to be the conductile cells. The conductile cells have one role, they have one job, and that job is to generate and propagate electrical signals. They don't generate any force. They are incapable of generating force. They pretty much have a degenerate cytoskeletal and membrane system to generate force that we saw in skeletal muscle. So they pretty much lack all that machinery. They have a little bit, it's kind of left over, it's kind of vestigial. But they have some, but it doesn't generate much force. Their job is to generate and propagate electrical signals. They generate and propagate electrical signals to this cell type, the contractile cells. Now, when you think of the myocardium, recognize that something like 99.5 or more percent of the cardiomyocytes in the myocardium will be these contractile cells. The function of the contractile cells is to generate force. These are the ones we've been talking about with the myocardium forming the wall of the heart. These are the ones that are generating the force that will provide the pressure, provide the energy to open the aortic valve on the left side and the pulmonary valve on the right side. They are the cells that are providing the force that propels the blood through the pulmonary circuit and the systemic circuit. So these guys do the work. These guys tell the workers to do the work. Without the conductile system, there's no contraction. Without the contraction system, there's no force. So you need both of these systems working in concert. One to generate and propagate electrical signals, the other now to use the electrical signals to generate force. And we'll talk about these two cell types. We're going to see that these two cardiomyocytes have a lot in common, but there's some real differences as well. When you look at these cardiomyocytes, first thing that strikes us is that they're uninucleate. They only have one nucleus. And remember, contrast that with skeletal muscle. Skeletal muscle may have several hundred, maybe as many as 1,000 or more individual nuclei scattered along the length of the cell. And each individual skeletal cell is one gigantic cell. That's not the case here. When we talk about cardiomyocytes, each cell has its own nucleus. Each cell, now we'll see, each uninucleate cell is connected to its neighbors by gap junctions. But again, this is the same statement as we have up here. The pacemaker cells set the rate and rhythm. So your heartbeat is sitting there as you're resting quietly, 60, 70 beats per minute. That's being generated by these conductile cells. The conductile cells are the guys that are setting the rate and rhythm. They have a particular signal, have a particular kind of action potential we'll talk about that sets the rate and rhythm. They're then sending information to the contractile cells. The conductile cells are delivering electrical information to the contractile cells to tell the contractile systems to contract. So we have basically two functions of this conductile system. One is to set the rate and rhythm, and the second is to deliver that electrical signal to the cells that are going to do the work. Now, in order to understand that, we have to spend a little bit of time on the anatomy, and I'll try to describe this anatomy as best I can. And I'm going to talk about a few major players here. The first one is known as the sinoatrial node. Now, when you hear the term node, what you should think of is it's going to be a cluster, or a bundle, or a collection. Collection is probably the best word. A collection of conductile cardiomyocytes. This collection of, this knot, if you will, of conductile cells in the sinoatrial node is almost always, there's some exceptions, but 99.99% of the time as you're sitting there, your heart rate is being started by the pacemaker cells in the sinoatrial node. This collection of cells in the sinoatrial node is then connected to another set of cells in the atrioventricular node. Now, an upshot of this that I'll try to emphasize when I do the drawings is that there's no electrical continuity between the atria and the ventricles. The electrical continuity between the atria and the ventricles is all carried out by this conductal system. And the first hallmark, the first structure that we're going to talk about is the sinoatrial node. That will connect to the atrioventricular node. The atrioventricular node then will send out these bundles of conductal cells that form the bundle of His. It's a single bundle. The bundle of His then branches into a left and right bundle branch, which goes to the left ventricle and to the right ventricle. But they don't go directly. The left and right bundle branches are these two big branches. But they branch and branch and branch and branch into these tiny branchlets called Purkinje fibers. So we're going to follow the electrical signals. The electrical signals will be initiated in the sinoatrial node. They'll propagate to the atrioventricular node. They'll be regenerated in the atrioventricular node, propagate to the bundle of His, to the left and right bundle branches, finally reaching the Purkinje fibers, which are the characters that are going to deliver the electrical signal to the contractile cells. Now we'll see that as we work our way through this, that for both the conductal cells and the contractile cells, one of the hallmarks of their structure is that these individual cells at their ends have their plasma membranes interdigitated. They're going to interdigitate at places called intercalated discs. And when we look closely at the intercalated discs, we find that the intercalated discs will connect these cells. Whether we're talking about conductal cells or contractile cells, at the intercalated discs, individual cells are linked by gap junctions. So those gap junctions are going to provide electrical continuity. So all the system, from the conductal system down through Purkinje fibers into the contractile cells, all of those are connected by electrical synapses. But we'll see that the sequence of events are going to be electrical signal starts here, goes here, 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 and finally to the contractile cells. Then we'll spend a little time talking about the electrical signals. The electrical signals in the pacemaker cells have a particular kind of action potential, but they're unusual. We haven't seen action potentials like this before. First, the depolarization is going to begin with something known as a ramp potential. It's kind of like a generator potential. But in the pacemaker cells, these ramp potentials are produced by a special kind of channel called an HCN channel. That HCN is an abbreviation. It means hyperpolarization activated cyclic nucleotide gated channel. There's meaning in that term. Tells you that those HCN channels take two forces to open them. First, they require the force of the hyperpolarization. But the second thing is they require cyclic nucleotide. Remember CAMP that we talked about in the endocrine system. CAMP reappears here. And CAMP will bind to the cytoplasmic side of these HCN channels. And if there's enough CAMP around, we'll open them. We'll talk about that. But these two things have to work in concert. You have to have both the electrical hyperpolarization and the cyclic nucleotide in the cytoplasm to get those channels to open. And then we'll see that the contractile cells have a separate kind of action potential. The contractile cell action potential is dedicated to delivering calcium to the contractile cells have a TSR junction where we have an action potential arriving at the TSR junction, causing the release of calcium. That calcium then causes contraction. Now, we're going to separate our worlds here pretty much into pacemaker cells have these action potentials, contractile cells have these action potentials. We'll see how they're different. But we'll see that as we move from the pacemaker cells truly up here in the sinoatrial node and the atrioventricular node, as we move from these sites into the bundles of His and left and right bundle branches, that the makeup of the channels producing these action potentials begins to change. That we move from the specialized pacemaker cells to things that look much more like what we're going to see as a sodium action potential. So we'll see this as a transition. But we'll walk our way through this, and I'll try to explain that in some detail. But let's begin with where we were last time. I think this is an important summary of what we were talking about in Friday's lecture. And I like this figure because it says a lot. The right-hand side shows the anatomy. And you remember we said that the two coronary arteries that feed all the blood to the myocardium, those coronary arteries originate at the base of the aorta. And that when the muscle's contracting and blood is rushing by these openings, we don't get much flow down to the myocardium. That's shown here on this graph. So on the ordinate, you see coronary blood flow. And on the axis, you see systole and diastole. And you'll notice that during systole, there's not very much blood flow. And that's not surprising, because remember, we said that the myocardium is contracting. When it contracts and those sarcomeres shorten, they're going to pinch off. They're going to squeeze shut those tiny capillaries in the myocardium. Because they're squeezing shut the capillaries of the myocardium, you just can't force much blood through those tiny capillaries when they're closed. So it tells you that during systole, which is the time that we normally think of pumping blood through the pulmonary and systemic circuits, because that's when we're generating force by those contractile cardiomyocytes. During that period, we don't pump much blood to the ventricular myocardium. So the question, of course, arises, where do we get the pressure? Where do we get pressure during diastole? Well, that's shown on the next slide. This is a slide we'll come back to when we talk about the vasculature, but I think it's useful to show it here. What we see on the axis are various sort of vessels. The ones I want to show you are basically the aorta and maybe a little bit the large arteries. But you'll notice that this is pulsatile here, that we increase the pressure up to 110 millimeters of mercury, and then someplace here at the peak, we open that aortic valve. The blood floods out into the aorta. But then the valve closes. The valve closes because the pressure on the aortic valve on the aorta side is greater than on the chamber side, but it closes. Because it closes now, we get the blood flowing through the system, so the pressure drops. Until we reach the point down here, this is diastole. This is typically the blood pressure you see. Average blood pressure is being 120, because we measure these in large arteries in the arm usually. 120 over, about 80, 75. But we're going to use this diastolic pressure. We're going to use this falling pressure in the aorta. That's the source of the energy to move the blood through those coronary arteries. So, if we come back to this, after the aortic valve is opened, and as long as they're compressing all these fine capillaries down here with the contraction of the myocardium, we just can't get much blood flow through those capillaries. So we wait until the pressure in the aorta has closed that valve. That's the beginning of diastole. During diastole, we retain pressure here in the aorta, and that's the pressure that's driving the blood through the coronary vasculature. And that's where we closed last time about the dangers of building up plaque on those coronary arteries. And Poiseuille's law saying you don't have to build up very much plaque before you start to seriously impede flow, because the flow is proportional to the radius raised to the fourth power. Tiny changes in that diameter or radius are going to have big changes on flow. So that's where we left last time. And so where I want to go now is begin to think about the types of cardiomyocytes we have in the heart. And again, I can't overemphasize, when we talk about this, don't think that the number of contractile and conductile cells are similar. They're vastly more contractile cells than conductile cells. But in fact, all of them, all the cells of the heart, I should say muscle cells, all the muscle cells of the heart break down into two types. One are the conductile cells, and we'll call them conductile conductile cardiomyocytes. And the other crew will be the contractile. Of course, these are the guys doing the work. But these are the guys who are instructing the guys doing the work what to do. So let's look at these conductile cardiomyocytes and talk about them in terms of what characterizes them. First, they are uninucleate. Secondly, they contain pretty much a vestigial contractile system. When you're looking at these cells in the electron microscope, you have to look very, very carefully to see the specializations. Of the cytoskeleton. Associated with contraction. You could abbreviate this pretty much by saying they just don't have much in the way of sarcomeres. You just don't see much of the actin thin filaments, the myosin thick filaments, the Z lines. Just is pretty much gone. And the second thing is that they lack, I should say lack. They lack specializations. They lack specializations of the membrane systems. Associated with contraction. So remember when we talked about contractile cells, we said they had this robust sarcomere system. We said that the contractile cells of skeletal muscle had a robust T-tubule and SR system. Well, you just don't see much of that in these conductile systems. That's pretty much gone away. But when we look at the contractile cells, and the contractile cells, again, they're uninucleate. And what we see is that they have highly specialized cytoskeleton for contraction. So they have sarcomeres. They're well organized. We see also that they have a well organized system of membranes. Specifically, we'll see that they have T-tubules and SR. All of this is to point out that these cells are organized to generate force. Just as our skeletal muscle cells are organized to generate force, so are these cells. But we also call these cardiomyocytes. Even though they don't do much in the way of generating force, they are cardiomyocytes whose function is to generate and propagate electrical signals. So keep those two ideas separate as we work our way through this. When you're here, conductile or pacemaker, that's this branch. And what they're doing is they're generating and propagating electrical signals, whereas these guys are responding to those electrical signals. But let's make a couple drawings. These are highly stylized drawings, but can take this and talk about the structure of cardiomyocytes. And their connections. So we'll start over here. And I'm going to draw one of these cardiomyocytes. They look a little like this. These finger-like projections of their plasma membrane at the ends. They have a single nucleus. This is a conductile cell. You could say it's a pacemaker cell. Now, from what we have in the upper panel, you know there's not much in the way of sarcomeres in there. There's not much in the way of T-tubule or SR systems. But we do find that each cell is connected to its neighbors at these sites where the extensions of the plasma membranes interdigitate. They don't fuse, but they interdigitate. And these structures, where the ends of the cardiomyocytes interdigitate one with another, has a name. These are called the intercalated disc. If you look carefully in the electron microscope to look at those intercalated discs, what you're struck by is that there are all of these structures called gap junctions. And these gap junctions provide electrical continuity between cells. Then, if we look at some of these, and I'm going to simplify the story, I'm only going to put a couple of them in. These cells, then, a subset of these conductile cells, will form gap junctions with the next type of cell, which is called a contractile cardiomyocyte. Now, I'll remind you that we're working down here at the cellular level. So we're talking about individual cells with their individual nucleus. What we see when we carefully look at the membranes of these contractile cells is that they have extensions of the plasma membrane. This is the sarcolemma. But just as we saw in skeletal muscle, the sarcolemma is continuous into this structure called the T-tubule. These T-tubules, as we saw in skeletal muscle, are closely associated with sarcoplasmic reticulum. So this is meant to be the sarcoplasmate. Now, whereas these green cells lack much in the way of cytoskeletal organization, when we look at these contractile cells, we're struck by the fact that there are these Z lines. And just as we saw in skeletal muscle, those Z lines are made of alpha-actinin, which is an actin-binding protein. So there are these actin-thin filaments. They're actin-thin filaments. The actin-thin filaments are attached to the Z lines, and between those actin-thin filaments are the myosin thick filaments. These myosin-thick filaments, as we saw in skeletal muscle, are poised to reach up and contact the actin-thin filament. And they will do so in the same pattern. That is, when the calcium is released from the sarcoplasmic reticulum, it binds the troponin C. The troponin C, with the calcium bound, moves the tropomyosin, opening those myosin binding sites on the actin so the myosin head can reach up, glom onto that actin-thin filament, and draw these C lines together. So the story of skeletal muscle contraction and the story of cardiac muscle contraction is very similar. We're going to see there's some subtle differences when we dive into this, but they're pretty much the same thing. But one of the differences between the heart and skeletal muscle is that in skeletal muscle, that skeletal muscle is driven by the lower motor neurons. It's the nervous system that tells that muscle to contract. That's not what we have here. In the heart, what we have are these conductal or pacemaker cells are setting the rate and rhythm of muscle contraction. So there's a division of labor. Division of labor among these fairly small number of cells where these guys are generating electrical signals. Those electrical signals are being propagated from one conductal cell to the next until finally they reach this interface between the conductal system and the contractile system. And then these conductal cells, through gap junctions, again, these gap junctions, the conductal system delivers electrical signals through gap junctions to the contractile cells to tell these guys to contract. So we have this kind of division of labor, and we have an anatomical division of labor as well. So any questions up to here? Okay, so let's take that pattern, take this up, and on this board, what I'm going to do is make another drawing of the heart, one of our sort of diagrammatic representations of the heart. And what we'll do is we're going to think about the surface of the heart. Now I'm going to draw this thing like I always do, and I'm going to show the chambers, but I want you to remember that we're talking about the surface of the heart here, just as we talked about the surface of the heart last time with the coronary vasculature. We're talking about the surface of the heart here. And I want to lay out the anatomy of the conductile system. I'm going to follow through that series of names over there. But we'll use our description here where this is the right atrium. This is the right atrium. This is the right ventricle, showing the walls. Now again, I'm showing chambers here, so I'm going to put the valves in. Now, if you were looking at the surface, you wouldn't see those valves, and we really want to be thinking about the surface. Over here is the left atrium, the left ventricle, the valve in between. But of course, we wouldn't see this. We were looking at the outside. And now, on this, I want to superimpose, without going into all the great vessels, I want to superimpose the conductal system onto this. I'm going to start up here, and I'm going to put a big green dot. And I'm going to label this as the sinoatrial node. It gets its name because it's right minus the region, a sort of a floppy region in the atrium, and this node atrium. So it's We commonly abbreviate this the SA node. And a node, in our parlance today, is a collection of conductile cardiomyocytes where the heart rate, I should actually say the heartbeat. No, I don't like that. Where the heart rate is initiated or determined. Well, we have this sort of knot of these conductile cells, but then they form sort of like a thread. And that thread will go out over the right atrium and go out over the left atrium. But extending from this bundle, this clot of cells, this knot of cells, is another process. It comes down to the ventricle. Put another knot in here. Now, one of the things I need to impress on you is that there's no connection between the myocardium of the atria and the myocardium of the ventricles. The only connection between the atrial myocardium and the ventricular myocardium is through this conductile system. If you actually look in here, there's basically a very thin sheet of connective tissue that separates those two chambers. So the only place where we can get communication between the atrium and the ventricles is through this conductile system. And so the next way station is here at this site, known as the atrioventricular node. Again, this is a collection of neuronal cell bodies. And so we initially talked about the SA node. And it's the SA node where the heart rate is determined. I think I just said that. But the AV node, the atrioventricular node, this node here, the AV node, is also rich in pacemaker cells that can set the heart rate. Typically, I think of the atrioventricular node as a kind of backup. If your SA node doesn't fire an action potential to deliver a signal, it can basically shrug its shoulders and. But under most cases, what's happening is the SA node sets up a signal. It sends that signal to the AV node. The AV node responds to that signal, and it sends signals out through another bundle here. This is a bundle of conductile cells. This bundle has a name. It's called the bundle of His. This is one of these eponymous names. Dr. His, I think, was a German physiologist in the 19th century. So we call this the bundle of His. But then that bundle of His, which is a single bundle coming out of the atrioventricular node, branches into two bundles. Into a left bundle branch and a right bundle branch. So we've set up the potentials almost always in the SA node, sends them to the AV node. The AV node then sends those potentials on through the bundle of His into the right-left bundle branches. But that's still all conductal system. We haven't yet delivered those signals to the contractile cell. So we haven't gotten to that interface that we show up above between the green cells and the red cells. The interface between the green cells and the red cells occurs after these left and right bundle branches further branch and branch and branch. And these are said to be the Purkinje fibers. Again, this is one of these eponymous names that's left over from the 19th century. Dr. Purkinje was a physiologist in the Ukraine. No, he was Czech, actually. Purkinje was a Czech physiologist. And Purkinje, these Purkinje fibers are the final arborizations of the final arborizations of the conductile system. That send electrical signals. Through gap junctions. To contractile cells. So we've divided our world into this conductal system and the contractile system, two different kinds of cardiomyocytes. The conductal system is further divided into these cells here of the SA node and the AV node, where we have pacemaker cells. But that's just electrical signaling. There's electrical signals that are set up in the AV node and in the SA node and sometimes the AV node, are then finally delivered to the contractile cells, and that signal coming from the conductal system to the contractile system tells those contractile cells to contract and generate force by contracting the cardiomyocytes in the myocardium, shortening the Z lines and generating the pressure on the chambers we talked about the other day. So questions to there. Let me show you a pretty picture of this. This is a much prettier picture than I have on the board, but it's the same thing. So what we're talking about here in this anatomically correct structure. So up here, that's the SA node, and it's showing these bundles of conductal cells branching out over the atrium. They don't show it here, but they'll also go from the right atrium to the left atrium. And then there's a single connection between this sinoatrial node and this connection here to the atrioventricular node. The atrioventricular node is a series of conductal cells. It's located on the ventricle, just about where the atrium and the ventricle meet one another. So that now becomes the atrioventricular node. The atrioventricular node then has a single bundle of conductor cells coming out of it. That's the bundle of His. Then the bundle of His will then separate into the right and the left bundle branches. And in those bundle branches, again, there are a bundle of these conductal cells. And they're all connected by gap junctions. And then these further branch and branch and branch, and these Purkinje fibers now are delivering the electrical signal. It was started up here in the SA node and propagated through the system. They're delivering the electrical signal to the contractile cardiomyocytes in the ventricular myocardium. That's how that system works. But what I want to do in the last few minutes is I want to think about the signals that are set up in those two distinct kinds of cardiomyocytes. So we go to the back board here. So we're going to start out here in the SA node. And I'm going to label this as action potentials of pacemaker cells. In the SA node. We could pretty much make the same drawing for action potentials in the AV node. It's just they're a little slower coming on. But there's a particular shape of this action potential I want you to recognize. So here's the graph. This is time. This is voltage. We've seen a lot of these before. This is going to be an action potential, but there's some functionalness to this. We're going to start out here, minus 50 millivolts, maybe minus 60. And there's this slow depolarization I'm going to label as a ramp potential. We'll see in a moment this ramp potential is dependent on a particular kind of channel called an HCN channel. But this ramp potential from the HCN channel permits the influx of sodium. And that influx of sodium is depolarized the membrane potential until we reach a point where we get to threshold for opening voltage gated calcium channels. So we have positive charge. The positive charge, we deliver positive charge to the membrane, and now we reach threshold, but now we generate an action potential. It's kind of a funny looking action potential. It only goes up to maybe 0 to plus 10 millivolts. Reaches a peak and comes back down again. Then we start over, another ramp potential. Reach threshold, fire one of these calcium action potentials, come back down again. Ramp potential. Now, as you're sitting there quietly, at say 60 beats per minute, this interval between the peaks of the two action potentials is about 1 second. A little less if you get up around 70 beats, 180 beats per minute. Now we're looking at 300 milliseconds between these. But, no, 30 milliseconds between them. So what I want to look at who these channels are. This channel here is the HCN channel. The HCN channel, which provides the depolarization that generates the ramp potential, the HCN channel is unusual. And the peculiarity of this channel is in the name. It's said to be hyperpolarization activated. So everything we've talked about before in terms of opening voltage-gated channels was a cation. But here we're opening with hyperpolarization with negative charge. So that's pretty weird. The second thing is that the HCN channel is opened by concentration. Of cytoplasmic. Cyclic nucleotide. In this particular case, the cyclic nucleotide is cyclic AMP. It's CAMP. The same signal we talked about in the endocrine system. Now, if we look schematically at one of these channels, we can think of it this way. If we look at a cross-section of one of these channels, you can see there's a channel in the middle, but there are 4 subunits. And each of those subunits has the capacity to bind to CAMP. The binding of the first molecule to CAMP to one of these subunits increases the probability of channel opening a little bit. Second, increases it more. By the time you have all 4 sites occupied, you're almost certainly going to open that channel. If you think about this across one of these membranes, let's go back up here. We can think about one of these proteins across the membrane, the pacemaker cell, but there's a subunit, there's a subunit, there's a subunit behind the plane of the board, and one that I've pulled out, so there's 4 subunits. On the cytoplasmic side of the membrane, we can now bind CAMP. Remember, this molecule is generated by adenylyl cyclase. See a little later, by activating adenylyl cyclase, we'll make more CAMP. The more CAMP we have, the higher the probability the CAMP will bind to these channels. The more of these channel sites that we have bound, the higher the probability this channel is going to open. This channel is permeable to sodium. And it's this influx of that sodium through that channel which is generating the slow ramp potential. The function of the ramp potential is to provide positive charge to slowly depolarize the cell until we reach threshold for the voltage-gated calcium channels. The voltage-gated calcium channels open, but then at the peak, these voltage-gated calcium channels close. And after they close, at this point here, we begin to open the voltage-gated potassium channels. That, of course, is going to repolarize the membrane. So that's the source of the hyperpolarization. And these are the two things working in concert. The hyperpolarization drives the membrane potential back down to, maybe 60 millivolts. That hyperpolarization contributes to the probability of opening the cyclic AMP gated channels. But the hyperpolarization itself is not adequate. You also need to have, working in conjunction with hyperpolarization, the cyclic AMP binding to these channels, increasing the probability of opening, allowing the sodium to come in. I think you can see from this that if we let more sodium in through the HCN channel, what's that going to do to the rate of rise of this ramp potential? If the more sodium is coming in, it's going to more rapidly depolarize the cell. So rather than having this nice slow slope on this thing, now with increased cyclic AMP activity, we more rapidly reach threshold. We fire an action potential more rapidly, come back down again, fire an action, let me put a little bit of a ramp in there, and fire another action potential. So when we increase the cyclic nucleotide concentration in the cytoplasm, we'll increase the rate of rise of the ramp potential. By increasing the rate of rise of the amp potential, we'll more rapidly reach threshold and increase the rate of this pacemaker cell firing. When that pacemaker cell fires an action potential, it then begins the process of propagating this potential down to the AV node. It's regenerated in the AV node because this is a bundle, this is a collection of conductile pacemaker cells. That action potential then propagates through the bundle of His, left and right bundle branches, and then down to the Purkinje fibers to deliver the electrical charge to the contractile cells. Now, a question came up in the first lecture, so I'll repeat it here. It's a really important point. These action potentials that we see here are truly in the pacemaker cells. But when we begin to look at the contractile cells, we see a different kind of action potential. So in this half of the board, I want to draw it in red, one of these contractile cell action potentials. When we map this out time, and on this we have voltage. The membrane potential sits at about -70, so it's more hyperpolarized. And at this point, we have the arrival of signal from the conductile system. This depolarization opens voltage-gated sodium channels. These voltage-gated sodium channels immediately depolarize the cell up to about plus 20 millivolts. Here they inactivate. And from there, we now have open for some period of time voltage-gated calcium channels that deliver calcium to initiate contraction. We'll start there next time, and we'll see that as we move through this conductile system, we leave behind these action potentials so that pacemaker cells and more take on these